Welcome everybody to this live webinar we are offering today for you. My name is Luis Valiño. I will be the moderator today. And with us, there is Noemi Ribes. She's our R&D director and the head of the oncology area at Bioarray. So Noemi will run the presentation first, and in the end, we will have your questions. For the questions, please, you can type them in the chat area of the webinar system. This is in the right bottom corner of your screen. Please look for that. And you can type the questions at any time as they come up to you. Then at the end of Noemi's presentation, we will just read them out, hopefully all of them, if we have time, and she will answer one by one, right? Mm-hmm. Then, let me just pass the first slide. I want to mention that obviously the purpose of this webinar is to share our experiences with this, with today's somatic mutation panel. But since we are a service provider, in case you wish to know more details on how we provide this test, for instance, sample requirements, how we collect the samples, how the reports look like, other tests available in our portfolio of the oncology testing menu, then you can just hit that blue request button, and then you will be able to arrange a short call with either Wasila Buddha or myself. My colleague Wasila takes care of North of Africa, Middle East, and I take care of the companies that are or institutions that are based on Europe and Asia. Okay, then you can minimize this offer panel if you want to have the maximized chat panel. You can play with it a little bit to feel comfortable. And now, before starting, just brief word on our speaker, Noemi Ribes. So after graduating in biology, Noemi obtained her PhD in 2016 at the Institute of Neurosciences. This is within our National Research Council. There, she worked as a research position, of course, with tumor suppressor genes involved in the development of glioblastomas. After that, she took a postdoctoral research position at the University of Michigan. For 4 years, she worked on stem cells and cancer genetics through multiple approaches, from transcriptomics to epigenetics. And nowadays, she's with Bioarray as R&D director and head of oncology, as I said, with a mission to offer the most advanced and reliable solutions for the genetic profiling and diagnosis of cancer. So without further ado, Noemi, please, over to you.
Hi, hello. Thank you for being here today, and thank you, Luis, for that kind introduction. So again, my name is Noemi Ribes, and as Luis said, I'm the head of Cancer Genetics Department here in Bioarray, and hopefully today I'm going to introduce you some of the solutions that we are currently offering to try to assist oncologists and medical specialists working in cancer to try to guide a diagnosis and also treatment selection. So there we go. So as all of you know, and it's something that is well established, cancer is a genetic disease, meaning that the changes that lead to tumor formation actually happen in our genetic material. Then it makes sense that we put so many efforts in trying to identify those changes in the DNA that actually going to help us to understand how a particular type of tumor might be behaving or might be developing, and also the possible targets that we can use to guide a successful diagnosis and treatment selection. And it was actually the potential of molecular genetic tools was initially recognized by hemato-oncologists who determined that the particular translocations were useful to identify the diagnosis or to guide the diagnosis of various leukemias and lymphomas. So first of all, what is a molecular assay? A molecular assay actually consists of an analytical test that is going to help us to measure molecular traits that are commonly known as biomarkers, in this case, cancer biomarkers. And as you can see here in this figure, we work with different types of biomarkers. that they can have slightly different meaning or slightly different use. So we have diagnostic, prognostic, and predictive biomarkers. The diagnostic biomarkers can help us to confirm the presence of a cancer or even narrow down the specific genetic mutation that is causing a particular type of tumor, or sometimes they are even enable or helping us to this early cancer detection. On the other hand, the prognostic biomarkers might help us to inform on the likelihood of a particular patient health outcome. And finally, these predictive biomarkers can help us to match the use of available target therapies or immunotherapies or even clinical trials to those patients who actually are going to benefit from the use of them. And just to give you some examples of these powerful biomarkers that we have been using for the last years in cancer genetics and oncology, I wanted to show you some of them, the three most prevalent cancers, which are breast cancer, lung cancer, and colorectal cancer. And as you can see here, over the decades, we have been able to discover many of these genetic biomarkers that are continuously helping us to narrow down the diagnosis and also to select the best or best fitted treatment for each patient. So on the upper left side, you can see a little bit of the map of the breast cancer situation. And in this situation was actually immunotherapy, immunochemistry, was first adapted for the clinical determination of the level of expression of estrogen receptor more than 30 years ago. And this was truly a historical advance in personalized medicine or personalized oncology, as it changed the medical attitude towards one of the most common oncological diseases, as it is the breast cancer, by tailoring or by helping us to tailor the endocrine therapy to those patients who actually will benefit from it. So the analysis of the expression of the estrogen receptor in breast cancer tissue, it demonstrated that only this ER-positive breast cancer patient actually benefit from estrogen ablation. Also, another example in breast cancer was the discovery of HER2 amplification that eventually led to the development of several HER2 target therapies with trastuzumab, for example. And from the really beginning, the administration of trastuzumab was tailored to women whose tumors show a specific HER2 amplification or gene amplification. And yeah, just to put another example, the well-known BRCA1 and BRCA2 are two genes that are commonly evaluated in breast cancer, and actually the mutational status of these two genes can relate or can help us to consider the administration of PARP inhibitors. On the upper right side, in this case, we can see a little bit more information regarding to lung cancer. And as you probably already know, lung cancer is the world's most deadly and prevalent cancer. But luckily, nowadays, we count on several genetic biomarkers that try to tailor treatment to those patients in order to get the best benefit and overall survival for this patient. And it was in the year, I think, 2004, that 3 independent research groups were able to report that patients responding to a particular treatment, that was the TKI inhibitors, where the responders were the ones that were carrying mutation on the EGFR receptor gene, versus the one that were not responding to that particular type of treatment, that were EGFR wild type. In the same line, early crizotinib clinical trials coincide with the discovery of ALK translocation in lung cancer. So the analysis of the drug responders again revealed that the presence of a translocation in this ALK gene was a prior prerequisite for the drug efficacy. And yeah, subsequently, studies or similar studies demonstrated something similar for another kinase, which is ROS1, which is also recurrently rearranged in lung cancers. And ROS1-driven tumors represent another category of tumors that are actually crizotinib responder cancers. And nowadays, ALK1 or ROS1 rearrangements are almost routinely tested in lung adenocarcinomas. And the list goes on and on and on. I can show you many other examples, with just to just to end up with another prevalent cancer, I'm going to mention some of these biomarkers in colorectal cancer, and actually the genetic status of also the EGFR receptor has also been proven to be relevant in colorectal cancer. So in this particular case, clinical trials working with antibodies, monoclonal antibodies against EGFR and receptor, particularly cetuximab or panitumumab, convincingly demonstrated that these drugs do not render benefits for tumors that are carrying mutations in other genes, which are KRAS or NRAS. And this is because these RAS proteins are actually part of the EGFR pathway, but they are downstream of the receptor. So in this particular type of colorectal cancers, which are carrying mutations of those genes, which are actually more than half of colorectal cancers, the overactivation of that route is completely independent of the state of the EGFR receptor, meaning that they are not going to be responders for these antibodies against EGFR therapy, and meaning that we need to move these patients to a different approach. So yeah, and as you can see, this is just an image showing the timeline of how we keep on developing not only, or we keep on describing new biomarkers in different types of cancers, but also we keep on discovering new drugs. And yeah, what we are witnessing is basically that over the last 2 decades, it's been an overwhelming advance in biomarker discovery and target therapy matching to those biomarkers. And nowadays, there are continuously new clinical trials testing target therapies in different cancer scenarios and trying to understand which are going to be the patients that are going to really get the maximum benefit with a particular type of therapy, depending on their tumor profile. And at the same time, international regulatory agencies are continuously approving new drugs. As you can see here, over the last years, having a massive approval of new drugs for various type cancers. And basically, this tendency is not slowing down. Clinical research, translational research, and ongoing clinical trials every day even more, and on the works new clinical trials together with us, with these regulatory agencies, as I said, are actively working to be sure that we are describing meaningful targets and to tailor the best treatment approaches to the particular tumor profile of every patient. And this is important because, as I'm saying, the point in here is not to be able to develop a new drug, which might be really good for cancer. The point in here is to be able to identify patients that actually are going to benefit for that therapy, for that approach, and that depends on what is the tumor profile of that patient. So because of that, molecular profiling is becoming a standard practice for most patients with advanced disease, where there is no time to lose, replacing the historical treatment paradigm or the classical approach, which was prescribing chemotherapy based on the tumor organ of origin or histology or the state of the cancer. And this paradigm is actually changing because there are everyday new clinical trials and new reports and new papers and new analysis showing that actually this target therapy tailored to the patient profile is working, is getting a much more better results in patients. And in this slide, I just wanted to show you one of these many meta-analyses that are particularly showing this. This is just an example where a comprehensive analysis of phase 2 clinical trials was developed to reveal across malignancies how were responding patients that were administered a tailored therapy depending on their tumor profile versus the ones that were not going through the personalized approach. And as you can see on the graphs in the upper part, this personalized approach, which is the two bars that we can see always on the left side, compared with the non-personalized approach, which are the two bars on the right side, consistently and independently correlated with higher median response rate, as we can see in the first in the first graph, with a much more prolonged median progression-free survival, the graph in the middle, but also with a better overall survival, the graph on the right. And okay, so I guess that now that we have more or less clear that it's really important to get this tumor profile from the patients in order to be able to tailor the therapy to them to get the maximum benefit. So the next question that we have to address is how actually we go over this tumor profile, how we assay the genetic profile of a patient. And you know that there are many different techniques for genetic testing. We can go for immunochemistry, karyomapping, FISH, microarrays, Sanger sequencing, MLAPA, or a much more new approach, which is next generation sequencing. And I'm not going to go with detail over them. I just want to point out that there are, for many of them, particularly the same type of limitations, which are that in the majority of the cases, not a great amount of genes can be tested, or not a great amount of biomarkers can be tested at the same time. And also the DNA input in many cases is too high or consistently high. So the way to go is two different approaches. We can go through the sequential approach, which is the image that you can see on the left side, or we can try to go over a parallel approach, which is the image that you can see on the right side. So multiple genes actually can be tested and applying this sequence of different tests over the same sample by using laboratory developed approaches or commercially available kits. And this approach, which is the sequential one that I was mentioning, is followed actually for many pathology labs in the world. However, it's important to mention that there are some limitations or some at least considerations to take into account if we decide to go this route. And the first of all is that because we are using different techniques to evaluate different biomarkers, every single technique has to be validated. So we have to be sure that we are doing the things properly and we are getting the proper results that we will expect to get from that technique. Also, and what in my point of view is sometimes the most limiting situation in cancer patients, is the limited amount of sample that is available. Following this sequential approach, we keep on needing extra DNA material to run the next test. And sometimes this is not possible because the amount of sample is really limiting. So the doctors in those cases have to actually decide which is the test that they want to run instead of going and be able to test more than one or more than two biomarkers. Also, all of these, as you can imagine, relates to additional cost, additional reagents that are needed, and for sure additional human resources, additional people involved in the testing. And as you can imagine as well, all of these might lead to a delay in the diagnosis or in the treatment assignation if we keep on getting negative results. And finally, also an important thing that sometimes is not considered is that different techniques or different tests are going to need different laboratory equipment and also different or sometimes different qualified experts to be able to run the test, but also to be able to interpret the data coming from that test. So there are several considerations that we have to take into account when we are deciding how to approach this molecular profiling. An alternative approach, as I was saying, is to go over a parallel approach to the molecular analysis. And this is based on the utilization of multi-gene NGS cancer panels. Next generation sequencing allows for comprehensive description, not only of germline DNA, but also analysis of somatic mutations and also RNA profile all at once. So this next generation sequencing allows sequencing multiple genes at a very high depth and coverage at the same time. So we are kind of somehow overcoming the limitations or many of the limitations that I just mentioned. One of the most important things is that with one sample, we don't need to decide. We can test many different biomarkers at the same time and get a really comprehensive profile of our patient, not being forced to decide because the amount of sample is really limited. Also, We don't need extra material, extra reagents. Everything is run with the same test, with the same lab equipment, and normally the people implicated is the same over the process. And as I said, we don't need to wait for the next result to be the answer. In once and with one sample, we can get the full profile of the patient. So this makes clear that precision medicine increasingly required or is requiring every day more these multi-gene characterization panels, and these are kind of becoming crucial for molecular analysis of the patients and the lesions present in those patients. So with that in mind, this is here in Bioarray, I will say that we are one of the first and also limited laboratories that we have the technology, the lab equipment, and the analytical capacity, which is also really important, to perform genetic studies of tumor samples using one of the most complete and updated multi-gene panel on the market, which is this one that you can see here, the Cancer Comprehensive Assy Plus from Thermo Fisher Scientific's OncoMine line. So as you can see here, on the right side, you might be able to already see some of the big highlights or the key highlights of this panel. So this is a really impressive panel, including more than 500 of genes, unique biomarkers, not only single gene biomarkers, but also multi-gene biomarkers. So as you can imagine, it's a very comprehensive and big profile for the patient, and we are able to detect all the type of lesions. We detect single nucleotide variants, indels, also changes in the number of copy of the genes, exon skipping, which is becoming interesting for a particular type of genes, but also because this test not only uses DNA, but also RNA input, we are able to check at once also fusions, not only known, but also novel fusions and splice variants. And another key feature of this panel is that, as I said, we do not only analyze single gene biomarkers, but also multi-gene biomarkers, as are the tumor mutational burden and the microsatellite stability, which both of them are becoming really interesting points to consider, where immunotherapy approach might be a consideration for the patient. And I'm going to go later a little bit with more detail, but I just wanted to show you that with this panel, we have a high coverage of the DNA repair pathways, and also we include the most important and prevalent biomarkers for the majority of solid tumors. So basically, with one panel, we can do a huge profile or a very big profile of the patient independently of the tumor that comes. We can test almost every single solid tumor. And one of the things that I wanted to mention that I consider also a strong hat for this panel is the fact that we require a really low amount of input to perform or to run the test. And as you can see here, actually the Thermo Fisher Scientific OncoMine line is the one that required the less amount of DNA to actually perform the samples. With basically 20 nanograms of DNA, we are able to perform this panel that I just mentioned. And this is extremely important in many cases because sometimes we receive really limited amount of sample, and so far I can tell you that we have not had to reject any sample. We have been able to successfully run the test and identify the biomarkers, and this might be critical for some patients where they are actually running out of material and they need to know at once what are all the options that they have. So, and as I said, this is really competitive with other tests available in the market, as you can see here in this upper table. Here on the bottom side, I just wanted to give you some insights into the assay performance. And as you can see, we are working with great sensitivity and specificity for all the type of genetic lesions that we are testing. And again, I'm not going to go over all the 500 targets that are tested here, but I just wanted to mention some things that might be really important for you to know, which is that we cover, as I said, more than 500 of genes, but these genes are being carefully selected, including the key cancer driver genes such as EGFR for sure, BRAF, KRAS, NRAS, ER2, or MET, which are some of the biomarkers that I just mentioned on the previous slides. And also, as I mentioned in the previous slide, there is a high coverage of genes involved in the DNA reparation pathways, not only the homologous repair pathway, including BRCA1 and BRCA2 and all the partners, but also the mismatch repair pathway. They are all really well represented here, and we are able to test many of the genes that are involved in those DNA reparation pathways. And again, we don't leave behind the RNA assay. We include the RNAi, and we are able to test these rearrangements, these fusions, including really well-known genes as ALK, ROS, RET, or the new all the NTRK fusions, which are again becoming a really interesting part of the fusions. And I'm sorry, I feel, I'm seeing that my camera went black. Sorry for that. I don't know what just happened. And I guess it decided not to go back again. Sorry for that. I will keep on trying, but it seems that has gone black. Luckily, or hopefully you still can hear me, right?
Yeah,
Okay.
no, I mean,
Sorry for that inconvenience. I don't know what happened there. So I'm gonna keep on going. So I guess the most important is that you can see the slides more than my face. But anyhow, again, this is just to mention what I just mentioned, that each genes, this is a really massive assay, and it does not mean that genes have been included in there just to add up. They are actually really carefully selected to be sure that we are covering the very most meaningful targets for every kind of solid tumor that we want to test. And they have been carefully selected from databases and coming from research literature, and also coming from the industry, considering the new pharmacological approaches that are being developed. And this is just to go a little bit over more details about the multiple gene biomarkers, because I consider it's something that is becoming an increasing point of interest, and these tests also consider these two, which are the tumor mutational burden and the the microsatellite instability, as I mentioned before, in order to consider immunotherapy. And this is because immunotherapy has been explored in the last years, proving to be really a very promising option across many solid tumors. And actually, the PD or PDL antibodies can induce a durable remission in solid and hematological malignancies. But however, only the 10 or 20% of them selected patients actually end up responding to this. So again, this is going over the same idea. The point in here is not keep on developing new new drugs. The point in here is not only to keep on developing new drugs, but to be able to match these drugs to the patients who actually are going to truly benefit from them. And for example, for the microsatellite instability, what we have is that a deficient mismatch repair pathway tumor is a tumor that accumulates thousands of mutations. And these particularly normally cluster in regions that are called microsatellites, which are consistent of repeats that might change on the length of the repeat. And actually, if this is altered, the length is altered, is when we observe this instability in the microsatellites. Therefore, a microsatellite instability tumor is a marker for a mismatch repair pathway, which is deficient or is not working properly. And which is really, really cool about the microsatellite instability was that it was the first biomarker to lead to tumor-agnostic approval of immune checkpoint inhibitors as pembrolizumab in 2017, after clinical trials show favorable responses to the PD-1 blockage in solid tumors with these high levels of microsatellite instability. And this approval was based on data from 149 patients that they showed or either high microsatellite instability or mismatch repair pathway deficiencies. And this was done over patients with several types of cancers. Actually, there were, like, if I don't remember bad, were 90 patients that had colorectal cancer and 99 patients that were diagnosed with other 14 different types of cancers. And as I mentioned, the ones that were showing this high microsatellite instability or deficiency in the mismatch repair pathway were the actual ones that were rendered benefit from this blockage, this immunotherapy. And kind of same situation here with the tumor mutational burden. The tumor mutational burden is defined as the number of somatic mutations per megabase that a tumor specimen might have, and this approximates the burden of immunogenic neoantigens that might be produced by a tumor. And similarly to what happened with the microsatellite instability, currently the FDA have granted approval of tissue-agnostic pembrolizumab for tumors showing a high tumor mutational burden. And in the clinical trials that the FDA based its approval was the keynote 158, nearly 30% of the patients with high tumor mutational burden had a response to the treatment, including those whose tumors completely disappear. And in contrast, only the 6% of the patients that they do not show this high tumor mutational burden show any benefit from the immunotherapy. So this tumor mutational burden and microsatellite instability have emerged as multi-gene biomarkers that are clinically relevant for a number of metastatic solid tumors that are treatable with cancer immunotherapy, and they have no other options. So they have joined the system portfolio of available tools for the personalized cancer treatment, and they are for that really important to take into consideration in the future. So again, here in Bioarray, we try to offer solutions for cancer genetics assessment based on next generation sequencing in order to assist the diagnosis and the treatment selection for cancer patients. And our goal is to work to offer an integrated solution for tumor profiling, and we always try to deliver the clearest and easiest to interpret results about the mutation landscape in each sample to guide the best fitted treatment recommended by the regulatory agencies. And all information that we provide keep constantly being updated with new discoveries, new publication regarding cancer research, but also we consider the new insights coming from the industry or pharma companies. And in the next couple of slides, I just wanted to give you a peek of how we are, how is the performance of the analysis that we are doing. So as I hope that you can see, this is a sample and we are getting a high amount of reads for the sample, being all the reads on target, which is really, really important, and getting a huge coverage over every region. If you cannot see clearly, I'm talking about the numbers on the upper table. So we get a high coverage of every region, and also the uniformity of the sample is amazing. So we are truly sure that we are performing a really good analysis, and all the targets are getting really well covered, and the uniformity of the sample is really good. So there are no biomarkers that are being well covered and other ones that might get not such a good amount of reads. So the performance is actually great. Again, even working with small amounts of DNA, it would be okay with 20 nanograms. And finally, I just wanted to give you another peek about how the reports look like. This is as you can imagine, a really comprehensive analysis, a really big analysis made over 500 genes. So sometimes the reports can be also kind of big and comprehensive. But what we always work on and try to do is to deliver the most important information in the first 2 pages. So the medical specialists, the oncologists, can get an overview of the situation of the patient and the options that this patient has in the first 2 pages of the report. So I hope that you can see it because the resolution doesn't look really nice, at least on my computer. But on the top part, you can see that the report is perfectly tailored to not only the patient but also the type of cancer that we are assaying. So on the first part, you are going to get the relevant biomarkers related to the particular type of cancer that we are analyzing. For example, this is a case for relevant colorectal cancer findings, meaning that you are going to get an overview of the biomarkers that are suggested or are normally in the medical guidelines recommended to be tested for colorectal cancer, and how is the situation for all of them, if we have been able to identify a variant, a pathological variant, or on the other hand, the biomarker is wild type or there are no relevant findings. So you can get an overview of the most prevalent and important biomarkers for that particular type of cancer. After that, in the next part of the report, you're going to get the whole information regarding to the biomarkers found and the matched therapy for each of them. You are going to get a matched therapy following the recommendations by the regulatory agencies for that particular type of cancer, but also we also provide a treatment recommendation for other type of cancers regarding that particular variant type. And again, the list of all biomarkers that you can see on the right side is all provided. All the findings are provided, being related to therapy in the case that there is therapy assigned, but also clinical trials for those patients that might benefit from the clinical trials. If there are additional findings, they are also included in the report. And if you go on, you can see much more details about this, as the allele frequency that they have been found. And in the following pages, for those of you that want to get much more insights or more details, you are going to be able to find the relevant biomarkers description. So you get an overview of the biomarker, where it has been found, and the relevance that has in different type of cancers, and all the details that you might want to check, but also much more detailed information related to the therapy that we advise in the report, where you can get all the details regarding to the agencies that they recommend that therapy and the usage, and all the details also related to clinical trials and the test itself. So again, as I said, a really comprehensive report where no information is left behind, but also well structured, where the most important information is always in the first 2 pages. And okay, so yeah, so with all of that, I just wanted to thank you, all of you, for taking the time to join us today. And hopefully I will have transmitted to you some of the new insights into these personalized medicines and the options that we have in the future for tumor profiling in order to help your patients to assign the best treatment option for them. Thank you.
Okay, thank you very much, Noemi. So let's take a quick look at the questions that we have written on the chat. Thanks for your questions, everyone. Yeah, now we can see you. Excellent.
Perfect. by the really end of the
we lost you for a while, but so yeah, let me start. We have a few of questions. Let's read the most important ones that we got. So first one, what proportion of the cases you have studied shown mutations in biomarkers linked to described therapies?
Okay, that's a really great question, because sometimes doctors might have this doubt. So I can tell you that for now, more than the 80% of the cases that, I would say more than 80% of the cases that we have been able to profile, we have been able to identify biomarkers that are directly related to therapies to follow. So the ratio where we find biomarkers that are actually actionable at the level of therapy is really high. Also, I don't want to leave behind the fact that even if other things, sometimes there are biomarkers that they are not included for any therapy at the moment, or not an approved therapy by the moment, but they are a consideration for clinical trial inclusions. And they are also really, really important because there are sometimes patients that they don't have other options, and these patients are getting also the information related to the clinical trials that they can benefit from and might be really useful also for those cases.
Very good. Then, what regulatory agencies' guidelines are considered for therapeutic assignment?
So we consider, or we follow the recommendations of all major regulatory agencies. Actually, in the report, you have the information, the treatment recommendations organized by the regulatory agency that is issuing that recommendation. But for sure, we include the FDA, the ESMO, the EMA, and the NCCN guidelines.
Great, thank you. Then, how many FFP slides are required to run the test?
So that depends on several things. For sure, depends on the amount of sample that. So the surface of the actual biopsy, the tumor that is included in the FFP block. So far, we do not have limitations regarding to the surface. We are accepting samples that come with a really small amount of material. The only consideration here that we actually request is to have a high tumor cellularity. So if samples have a high tumor cellularity, or at least a good tumor cellularity, the amount of tissue is normally not a problem. So to give you a number, normally with 2-3 slides maximum, we are working really nicely, and depending 2-3 of 10 micras are more than enough to work with the analysis. And sometimes it depends on the laboratory of the hospital. Sometimes we receive the whole block, we get the sample that we need, that might be even one slide. It depends, as I said, on the particular case, and we give the block back. And yeah.
Yeah, excellent. And then let's take the last one. Is it possible to contact with the geneticist to discuss information included in the report? I guess this refers to contacting our specialist.
Yeah, we, as I said, we always provide the maximum, not the maximum amount, the most comprehensive, but also important information that we can in the report. But always you have the option to contact us to discuss the information included in the report. If you have doubts about particular thing included in the report that you want to talk a little bit more in depth with us, I personally respond to those emails or conference calls. I'm always available, and we can have an open discussion in the case of the study if the doctor might want to have that option.
Very good. So I think we can wrap up now. Thanks a lot for the presentation and for answering the questions, Noemi. And to the audience, just thank you for your time. Obviously, there you have the main contacts at Bioarray for this topic, for our oncology menu. And then just I will leave for a few seconds after wrapping up the option of requesting a smart video call with the support team by clicking on the blue button in the offer panel. And that's it. Thanks a lot again, Noemi, and have a nice rest of the day to all of you.
Thank you all of you for spending the time with us. Bye.
Goodbye.